My name is Valerie Arboleda. I'm a pathologist, but I'm also a genetics researcher. I kind of live in both worlds, and I think this is the last formal talk of CGSI, so I'm going to keep it kind of light. I'll go through a good amount of background and sort of expand on some of the things that we're working on in the research space. So first, I'm going to start off with what is a pathologist, because I often tell people that I'm a pathologist, and then they're like, what is that? I've never visited a pathologist before. And that's actually a good thing that you've never visited a pathologist, because when you Google it, you get pictures of people who are looking under a microscope, just like Corey said, and they're usually physicians that do autopsies. So if you're the subject of an autopsy, that's never good. That means you're dead. There's bits and pieces. We look at tissue and cells under the microscope to diagnose cancer or inflammatory conditions. And the third thing that a lot of pathologists do that I think is the least known is that often pathologists are running clinical laboratories. So if you get a blood draw, if you do a nasal swab for COVID, it's all being run in what we call a clinical laboratory that processes these tests and makes sure we don't mix up your specimen with something else. And so I think about pathology, it's super fun because sometimes I speak with some of my older colleagues, and they will tell me that if they had known that there was a field like pathology where you could do a lot of research and have a lot of data without having to go see patients, they would have thought about pathology as a profession. And I always think about it as we have really exciting research tools. We build them in the research space, and they're super fun, and you can tweak them. And then by the time you bring it into the pathology lab, you've locked that thing down, and you are not changing it for like 20 years, because every time you make a tweak, you have to validate it, and it costs a good amount of money. So you might be tired from looking at like 5 million prostate cancer core biopsies, or sort of bored because you're doing the same thing over and over again. So I spend some time in the clinical space, about 6 weeks a year doing clinical sign-out, but I do spend the most of my time doing genetics research. And so for those of you, some of you might have visited the SWAB-seq lab. I spend a lot of time thinking about sequencing and genomic medicine, and I always show this slide. The sequencing is easy. It's actually one of the most straightforward things you can do, and I think it's very straightforward. It's about as straightforward as doing any of the other tests we do in the clinical lab. You can run it on a Illumina NovaSeq, which is here. This is actually the older version of it, the newer one. I just haven't updated my slide deck. Or you can run little nanopore things on your phone. I don't know how many people actually do this because I don't think you get much data, but it's possible. But interpretation is really hard. Like, how do we decide what variant is causing disease of the billions of variants in the genome? And with what certainty can you say this? Because you have to be pretty certain of things if you're going to put it on a piece of paper, give it to a patient, and tell them this is your diagnosis. So we get a lot, like I said, sequencing is easy. You can get like millions and billions of rows of different variants. It kind of looks like this, so no one's hopefully looking at this on Excel files. That would be crazy. And it kind of makes your eyes roll to the back of your head if you're trying to think about genetics related to phenotypes. And so we're going to talk today about Mendelian single gene disorders. And I think about these as they're monogenic, and so there's a single gene that causes the disease. Generally speaking, and this is all in generalities because a lot of the things I'm going to say here, there's always an exception to the rule. So they're rare in the population. They're not all super rare. You can have high effect size variants. They're necessary and sufficient to cause disease. That's not all, they don't always cause disease. And then they're protein coding, which is most of them, but there's a handful that we can find that are non-coding, and likely more as we go through, get more whole genome sequencing back. And they're typically identified by linkage classically and then with exome sequencing. So when you think about rare diseases, the FDA has a definition. It's something that affects less than 200,000 people in the United States. So that's sort of an arbitrary definition. It depends on how you're getting diagnosed and how easy that particular disease is to diagnose. But what we assume is that for any gene in which you know that mutation causes disease, we assume that they're, at least in the clinical space, we often assume that they're fully penetrant. So if you have this mutation, it causes disease in 100% of people, which is not totally true, and that phenotypes are the same across individuals with mutations acting by the same molecular mechanism. So if 5 people in this room share the same mutation, we should all have the same mutation in an LDL receptor. We should all have the same level of hyperlipidemia or of high LDL levels. That's the assumption we make in like traditional clinical genetics. If you're signing out clinical exome sequencing, we make these assumptions because most of those disorders are super rare. So we're thinking about things that are present in 1 in 100,000 individuals, very like pediatric, rare developmental disorders. But the question that we want to ask is, as we've sequenced more and more people, I think those 2 assumptions have really come into question. And we're starting to ask these questions about penetrance and expressivity of monogenic disease. What factors affect penetrance and expressivity, and to what degree are any of these genes fully penetrant, if any of them actually are? I will say that even for the most rare disorders, things where we find like 30 or 50 people in the world are diagnosed with this disorder, we're finding that there are individuals who get diagnosed clinically with very minimal phenotype or very atypical phenotypes, and the real question is why and what's going on. So I put this up here to show, just to demonstrate that this is maybe these like sort of hard definitions are fading a little bit into the background, and it's a lot more nuanced than we thought it was. Now, to give a little flavor of whole exome sequencing. So we do whole exome sequencing. It's been about almost 15 years where we've started doing it at UCLA, but there's a number of companies, GeneDx, Invitae, that do these panels and our whole exome sequencing. We and others have published many papers in this space. And the diagnostic yield, I was just talking with Lisa about this, hasn't changed. It's been somewhere between 25 and 40% over the last 10 years. And so what this means is your diagnosis fails 60 to 75% of the time. So we really have a lot of work to do and to try and understand what we're missing, and if it's, we've discovered everything there is to discover for true single gene disorders, and maybe there's a lot more nuance in multiple variants or other variant expressivity that we need to explore. So one of the things... So these are all clinical testing. So the diagnostic yield is if you have 100 patients, how many of them are you able to give a true genetic diagnosis, so a genetic mutation that matches their clinical phenotype. So I think about them as variants that are likely pathogenic or pathogenic based on a sort of a scoring system that's developed by the American College of Medical Genetics. So if you fail your this diagnostic yield, it means that you don't have a genetic variant that explains your phenotype. So all phenotypes, I think as you all know, exist on a spectrum. So I like the classic one of height where there's many loci. Most of us fall into the normal range, and then there's a set of individuals who are short and tall. And in these sort of extreme tails, you're more likely to have a rare variant that contributes to this particular phenotype. But we know that all of these phenotypes can be influenced by high effect or rare variants or common variants. So we kind of look at both of these things. And the question that we ask is, what are other explanations for these variable phenotypes and expansion of these phenotypes? So what other things are we looking for? So there's environment, which genetic testing usually won't take into account, but there's also common genetic variation, structural variants, and additional rare genetic variation. So another gene that maybe contributes or is in the same pathway as the initial gene. And so that is how we think about phenotype expressivity. I'm going to talk to you today about a project. I have a graduate student, Angela Wei, who is co-mentored by Noah Zeitlin, and we're going to talk about sort of exploring variant pathogenicity predictor methods against real-world penetrance and expressivity in the UK Biobank. And then the second thing we're going to talk about is sort of how genetic background modifies the expressivity of individual variants. I'm going to kind of touch on two things today, and here's how we've framed the argument for Angela, at least. So we have a phenotype that can vary in expressivity from low severity, high severity. We have genes, and then we have a population. And so each person in the population can have maybe a specific monogenic disease variant. So these are really well curated in that we took them from a small group of genes that have been established across multiple papers to be associated with the clinical trait of question. So for example, if you're an A variant carrier, let's see if I can. If you're an A variant carrier, you're fully penetrant for disease, so they're all the high expressivity, and that is very uniform. If you carry this B variant, it might be fully penetrant for disease, so you all have disease, but it might be sort of heterogeneous on how severe your disease is. And then there's C variant carriers. These are the ones that are by far the most challenging. They're incompletely penetrant and have a wide range of expressivity. And then we're going to look at a couple factors that are influencing this penetrance and expressivity. So you can have your PRS, so a polygenic risk score that influences the expressivity of all the carriers. So it has an effect in both groups, and it also influences or modifies the role of a monogenic variant. There's epistasis, and I'm not going to touch too much on that today, but that's a lot of work we've done with Sriram. And then we're going to also look at monogenic variants, and specifically we're going to focus around missense variants because they're the hardest to diagnose in the clinical lab, and they're the most common type of variant. But they're the most common type of variant, but I would say about half of them end up being variants of uncertain significance, particularly in genes that are clinically relevant. So I wanted to emphasize that if you have a disease-causing mutation, it doesn't guarantee that you have a phenotype. So I wanted to put these, I talked a little bit about penetrance and expressivity, but I want to put these two into the same slide so you can kind of just reinforce the difference. So I think about penetrance as something that's binary. Either you get the disease or you don't get the disease. And typically, because you can get them at different ages, we set a cutoff that's like by this age, some percentage of people will have the disease. So BRCA2 is a good example of this. 31% of people who have a BRCA2 mutation that's pathogenic will not develop breast cancer, while 69% of women will. Versus expressivity, which is more a continuous trait, and then if you think about FBN1, which causes people to either, if you have it, you can have a variety of variant phenotypes. You can either be tall and slender, scoliosis, you can have more moderate phenotype like scoliosis and joint mobility, hypermobility, or you can have a more severe phenotype that can be fatal, aortic degeneration and spontaneous pneumothorax. So for Marfan syndrome, you have this real spread, even though there are a couple classic mutations. So we really want to understand how many people are carrying these, who are asymptomatic and carrying these monogenic variants, and how does it change over time? So we've answered the first piece. The over time piece, I think, is going to be more complicated. But the real question we're trying to answer at the end of the day is, well, we have all these variants and all this clinical information. We're trying to figure out how you are going to address clinical variant carriers. If you diagnose somebody with a clinical variant, can we be a little bit more precise with our prognosis and trying to understand how you're going to tell people, if you're going to tell people they have a monogenic variant and it's only penetrant in 30% of people, is that something that people really want to know, and how to portray that information to others? So we looked at a handful of traits, and I know, at least in the Biobank meeting that was right before this, we tried to pick traits that were ascertained really well. So everyone in the UK Biobank gets measured for the cardiometabolic traits. Everyone has a height, everyone has a weight, and therefore you can calculate a BMI. So a lot of the traits that are uniform across the 200K plus release of the UK Biobank. We looked at a well-curated set of monogenic genes. So these genes are ones that have been very well established in the clinical literature to be associated with monogenic forms of hyperlipidemia, hypertriglyceridemia, and obesity. And then we looked across all of UK Biobank to figure out how many clinical variants there are, how many people are carriers of any very well curated clinical variants. So you can see here, we don't have that many clinical variants, and then this is the number of carriers we found in UK Biobank in the 200K release. And this is the UK Biobank that we used For this part of the study, we included everyone. We did not only use European unrelateds. We had to restrict for the polygenic risk score stuff I'll show you later. But for this part of the study, we just included everyone, all comers. So we first asked, what does the penetrance look like in the UK Biobank? And this is work that has been done in a smaller cohort of the UK Biobank, and we find pretty similar results. So what you can see is that there's almost nothing with a penetrance of 100%. Most things are somewhere between 50 and 75% penetrance if you have a very well curated gene. And I'm emphasizing that curated gene because it's only a small subset of variants within any of those genes. The vast majority of mutations or variants that you see in these genes aren't associated with the clinical phenotype. So you'll see them, and then that's where a lot of the questions come up because they come up as a variant of uncertain significance. But most things are not fully penetrant. Part of this might just be it's a snapshot. It's the time at which this person was ascertained, and maybe they will get there as they get older. But it's consistent with what we think with autosomal dominant disorders. Is there's a lot of things that can contribute to this. And the last thing I want to note is that if you're asking, did we account for statin use? We corrected for statin use in all of these individuals because that's practically in the water out there. So, okay, and then the question about expressivity. So of people who carry these well-curated monogenic variants in all of these genes, how variable are the levels of their specific trait? So you can see along the x-axis are the different genes we looked at. Each dot, it represents a carrier. And then along the y-axis are the different traits we measured. So we looked at high-density lipoprotein, low-density lipoprotein, and all the different hemoglobin A1C, BMI, and triglycerides. So you can see there's quite a range, and even between two genes that are monogenic for the same trait, you can see a pretty wide range of expressivity amongst people who are well-curated known carriers of monogenic pathogenic variants. Okay, so those are the well-curated variants, but what do we think about all the other variants that are out there, all the other missense variants that we all carry that could range in effect to nothing happens, to these could be pathogenic, but nobody's done the functional validation study to prove that that's actually the cause. And the thing that we did here is we leveraged this novel protein language learning model. It's called ESM1B. I think Vasilis Natranos, gave a talk on it at the very, very beginning of CGSI. And the question is, can we better classify the effect of missense variants for use in clinical care? So this is a problem that I come up with a lot. We see tons of missense variants, and we're like, oh, that could be a gene that we're interested in. That could be it. That could affect it. But we can't tell. There's no evidence in the literature. This is a rare variant, but should we saddle a person with a variant of uncertain significance if we have no evidence that it's associated with clinical disease? So we really want to identify these missense variants that have the highest impact of phenotype. We're not interested in sort of the ones that have small effects. We're looking at ones that are maybe moderate to high effect, that maybe push your penetrance up to another level. And some of these are not necessarily that rare in the population. So, sorry, this is not the right slide. So just because I know we're a lot of computational biologists here, I'm going to take you through what a missense variant is. So missense variants affect a single amino acid and change either protein structure or function. So if you have a coding strand of DNA on a protein coding gene, you can maybe have a change, which is represented here in the T. And this will be transcribed into a processed transcript. So you splice out this little gray intron. And then you translate it to make the actual protein, which is that functional piece. And each of these three nucleotides encodes a specific amino acid. And so just by changing an amino acid, you can have a variety of effects. You can change something to another amino acid that has the exact same biochemical properties and has no change in the protein structure or function. Or you can change it into something that has very different biochemical properties, changes the folding, and causes disease. Right? So this is something that happens in sickle cell. There's a very common glutamine to valine mutation at amino acid 8 of the protein. And that can cause this structure of the actual hemoglobin to form properly. And essentially you get what red blood cells that look like sickles. So this altered protein structure is sort of the final outcome, and then you have all the clinical phenotypes. But this is what pathologists might see under the microscope if you're looking at blood smears. And so we have, that's a very well classified variant, but there's a lot of missense variants that come up. There's more than 200,000 in ClinVar. And ClinVar is a database that's not well curated, so don't take it at face value, but it's a database where a lot of clinical labs will diagnose and put, they'll do a ton of whole genome sequencing, they'll put out diagnostic reports, and then they'll say all the different things that they identified they will put into ClinVar. They try to put into ClinVar. But it's not well curated because a lot of them are variants of uncertain significance. About half of them are labeled as VUS. And there are methods that aim to predict what the effect of these missense variants. And I would use them with caution because they often disagree with each other. There are ones that are supervised machine learning, so CAD, Polyphen, and SIF. Some of them have evolution databases built into them. And then there's unsupervised machine learning. And so these are more recent ones. I think Eve is one. There's Primate, I don't know why that's in bold, but Primate AIs, and they have a newer version, Primate AI 3D. And ESM1B, which is the one I think we're going to focus on today. Some of them are publicly available, some are not, like the Primate AI, which is Illumina's version. But ESM1B is a 650 million parameter protein language model. It was published initially in 2020. It's built by Facebook. I don't know the details of this because I didn't run the model. But the deep trend, it essentially takes a lot of the data from UniProt, UniRef, and uses a number of different parameters to do a prediction of all the different amino acids in all the different protein spaces. So there is a nice article in Rivas et al. Describing the initial pipeline, but then also a newer paper that's coming out. I think it might actually be out already. It was in press a couple weeks ago out of Vasilis's lab. And it's a really cool method, and it has all of these, the whole model is published. Not the model, the annotation is published online, and you can actually pull down all of the different annotations and look at your favorite variant in it. So we asked whether the clinical variants, the score of the specific clinical variants we're looking at actually corresponds with the clinical phenotypes we see. And then we did this in MC4R because we wanted to, there's a couple things. We wanted to know if they were associated, and then we wanted to know if it could distinguish between gain-of-function mutations and loss-of-function mutations. So loss-of-function mutations are things that just knock out the protein, and in this case, for MC4R, it causes obesity. It's one of the few high-penetrant monogenic causes of obesity. And if you have gain-of-function mutations, it's actually shown to be protective from obesity, and that's been mostly done in functional modeling. So the first thing we looked at was whether or not these missense variants can be predicted by ESM1B score. So I'm going to walk you through this particular plot because you're going to see a lot of plots like this. Along the x-axis, you have the ESM1B score. On the more negative the score is, the more pathogenic it is. So the more likely it's supposed, in this case, it'll associated with obesity is what we would predict. And then on the y-axis, you see mean BMI for an individual patient. Each dot represents a different variant. We could have made this plot and made the dots proportional to the number of people who were in that variant, but then it's completely uninterpretable. But no, there's more. There's at least 3 people or more in each dot, and then it's an average of the people in that dot. And then you can see here we have, so everything that's in gray, those are variants of uncertain significance. They have not been clinically annotated. If you see the ones in blue here, those are the ones that have been well curated and proven to be associated with monogenic obesity. The ones here in pink are the ones that are functionally validated to be protective against obesity. And then what you can see here, this red line, that's the threshold for obesity. And then this yellow line is the threshold for being overweight. So you can see there's this nice trend. We have a nice trend. This is 14,000 individuals and an R of 0.29. So it's reasonably good at predicting. It's way better than some of the other models. We validated this in BioMe, so this is just the first set that we sought to validate. It's a much smaller cohort, about 1,400 people. We see the exact same trend of the individuals who exist in BioMe with the variants, and then also the protective ones as well. I think what I kind of notice is interesting is the ones that are protective are still in the overweight and obese range, and maybe that speaks more to sort of lifestyle than anything else. But it was sort of an interesting reflection on society at this right now. Okay, and so we looked across a number of the point I wanted to make here is that none of the scores can even think about distinguishing between gain and loss of function that we've looked at. I've never seen a score that's been able to do this or do this even reasonably well. And I kind of went back and did a double take and said, well, maybe there's something else. Maybe the other ones do a better prediction, and I just have never really looked that closely. So we took a bunch of scores, and we took the same exact data, but we plotted them across the SIF score, the polyphen score. You can see that we always put the pathogenic range here in blue, and they don't correlate quite as well. You can see that the spread of variance isn't across the entire range of scores, and so that makes it very hard to interpret. And there were a number of prediction scores that we tried in which not all variants were included, so they would sort of cherry pick their favorite regions and only do predictions on ones where they had high confidence, and then the other regions were just ignored, like they maybe didn't matter so much. So this is CAD, both raw and the Fred annotated CAD. You can see in the Fred score CAD, everything is pathogenic, but it doesn't distinguish between these. We really think that ESM1B outperforms other variant classifiers for gain and loss of function, but just also in general, the correlation is much better with ESM1B. It's not perfect, but I think we're getting to that space where we can actually use it meaningfully for missense variation. Oh, and this is the Eve score. So you can see that there's almost nothing makes it up here. They don't have a pathogenic range, and they dropped a lot of our variants were dropped in this process. So the other thing we want to look at, so that's gain and loss of function. Just what about all the other traits that we were interested in? So we looked at a number of traits. So I'm going to switch to low density lipoprotein, LDL. And that's a trait that's associated with increased cholesterol levels. And then if they're high for a long time, you're often on a statin, and it predisposes to heart attacks. Now, it's the same layout, ESM1B score on the x-axis, mean LDL on the y-axis. And you can see there's a nice correlation. We don't have gain and loss of function anymore. So now your colors coordinate with the dark blue is curated high LDL scores, and the light blues are ClinVar. So the ClinVar that's likely pathogenic or pathogenic, and only that curation. So they're not curated to have if it's curated in ClinVar to have some that are likely path and some that are benign, we just didn't include it as a high confidence variant. So you can see here we see the same effect for LDLR. It goes in the more pathogenic your mutation is, so the more negative your score, the higher your LDL is in UK Biobank. And then when we look at PCSK9, which is a recessive trait and a protective one, the score goes in the other direction. It's not as nice of a correlation, but it's still there amongst our individuals. So everyone here with a variant doesn't even pass the threshold for hyperlipidemia. Okay, so ESM1B scores are better. I think that what I want you to take away from that is that we think it's better than some of the other curations. I think you'd have to test it across a lot of other genes and traits that are well curated. But I think this gives us a window. There's quite a range. Sometimes it predicts better for some genes than others. And I think we'd have to go back and do some nice functional studies. But it's sort of a window into how we can better predict missense variation, which has been a real challenge in the clinical genetics community. So the next question we asked was, does PRS influence the expressivity of a carrier phenotype? So amongst these carriers, you see this wide range of expressivity that I showed you earlier. Does PRS affect, is PRS contributing to that range we see amongst all the carriers? And so the answer is yes. It varies across the different the different traits. But here you have all the individuals who are carriers for MC4R. And then you have along the x-axis BMI PRS. So this is their scaled PRS score. And then you have along the y-axis again the BMI. And you can see that for all the carriers, as your PRS score increases, your BMI also increases. So there is a contribution of PRS to the expressivity of our monogenic carriers. We see the same thing in high LDL. So this is all of the individuals who are carriers for these same variants, and also for low LDL carrier variants, which is PCSK9. So is that right? This might be the wrong graph associated with that. But we see the same trend. I can double check that for you guys later. We see the same trend across most of our traits. It's not all, and the effect is a little bit it's nuanced. So some of them, for some traits it's really strong, and for some traits it's less. It has less of an effect. So it's really trait dependent, and maybe depends somewhat on sort of the strength of that polygenic signal. So we really wanted to think about how these common variants affect monogenic variant carrier phenotypes. We have here MC4R, which if you have a mutation, you are obese, and then also that contribution of common variants, or that PRS. So we know that these traits are influenced by common SNPs and environmental factors. And so I think what I'd like to kind of change the framework about how we think about any given trait is that there are some that are as a complex disorder. We think about them from the polygenic risk capacity, and that's pretty, that's a great way to frame that question when you're looking at common variants. But also as a monogenic disease, and how can we bring those two things closer together? Because at the end of the day, it's an individual who likely has some monogenic, some high effect variants, but also has some PRS score that you can calculate from all their common variants. And it's really a question of I think it's really going to be important as we move forward and more people are getting genetic testing. How can we integrate all these data sets? Not just those monogenic, so I always report out these exome cases where you have a single gene mutation. What about all the other variants that might be contributing? And then what about thinking about PRS and folding it into these models? So I think it's fair to say that PRS is going to influence maybe a lot more individuals as far as clinical phenotypes, but also once you bring in this question of moderate effect size variants, these rare variants that I showed you just before, you're going to see a lot of, it's going to be a lot more genetic testing, a lot more genetic counseling that is going to be needed. So I'm just going to show, I think everyone here probably knows polygenic risk scores, that it quantifies an individual's risk for a genetic disease. And the key things to think about here are we really care about the people at the tails of this distribution, so people who are in this high-risk variant scores. And so we wanted to just ask this question, like, how useful would any of these things be in the clinic? So if you're taking polygenic risk score, and this is across all of the individuals who are non-carriers, and then you look at the obesity carriers, at what point do you get to that threshold that's similar to a monogenic trait? And we've seen this in a lot of the cardiac traits before, and it really depends on where you draw your thresholds. But you can see here we have this curated version, which is like the variants that we know are associated with disease. And we've divided the ClinVar variants into weak ones that have sort of conflicting interpretations in the database, ones that are strong, or very high, strong in that they're consistent in the database, and then ones that are unknown, not in ClinVar at all, and have a low ESM1B score. Do we predict things pretty well? And what you can see here is that here's your PRS curve. It looks quite nice at the tail of the distribution. We're right there at the same effect size. These are high-risk individuals who look very similar to carriers of MC4R. And then if you take these unknown individuals that don't have a diagnosis in UK Biobank, many of them should be classified, we think, as pathogenic or likely path, given this information. We see the same thing in LDL, but to sort of or HDL to a different degree. So we have our curated variants and our ClinVar strict, which are consistent, anyone in the UK Biobank with an ESM1B score of less than 7.5, so the pathogenic, what we think are the pathogenic variants, the effect is less, right? So it doesn't actually get you to this higher LDL score. And so ESM1B is not sort of it's not a panacea, it's not perfect, but for some things we think it's doing a really nice job. And the same thing goes for triglycerides. We see a moderate bump for these unknown individuals, and maybe it will require a little bit more curation on our end to figure out where the right threshold for clinical variant interpretation is going to be. And this is the last one. This is the one that I think is kind of cool in that we have LDL, low LDL carriers. So this is the PCSK9 individuals, and then this is LDLR. And you can see that our predictions for the unknown PCSK9 is actually for the low carrier. So this should actually be should be above a higher ESM1B score. You can see that it's actually the unknown individuals aren't actually protected. So we can kind of write those off a little bit to say, well, these are not ones you want to say, oh, you're protected in some way from high LDL levels. And the individual, but what we see here for our unknown and uncarrier, we don't see a significant effect for these unknown ones. So we have to kind of toy. I feel like there's going to be a lot of work still to be done around finding that right cutoff. Right now we're using -7.5, but I think it might be disease specific or trait specific where you draw that cutoff for your interpretation. So this is pretty much the end of my talk. I wanted to really emphasize how it's really important to have a good variant predictor, because we're not going to do, at the end of the day, we're not going to do functional studies for every single one of the genes that are known to be monogenic. That would just be a Herculean task for any one lab or even a group of labs. And even if you could do all the different genes, the thought about doing every single missense variant is just a little bit daunting on the wet lab side. So we really have to think hard about how we can incorporate maybe some of the data that's out there with these pathogenicity predictors and improve these predictors to make them more useful in the real world. We have a lot of predictors, but most of them are, I would say, are garbage. They don't work that well, and we look at them, but we don't use them in clinical interpretation. And then the other big issue that I'd like to just make sure that you think about is how we can incorporate all of this data we get together, right? It's not just the genetics, the single gene variants, and the PRS. I mean, we heard a nice talk this morning about thinking about STRs and repeat expansions. Like, how can we incorporate that if they affect clinical phenotypes, and incorporate that into a way where, for an individual, I can actually give you an assessment of your genetic predisposition to disease, rather than 5 different reports saying you have this contributes 5% to your disease risk. That's not super useful, and I would say the vast majority of the general population wouldn't understand. And I think as we start to do this better, we're going to be able to do better prediction, because we need to put in more things to get to that ideal place where genome medicine will be looked at and actually be useful for patients and clinicians. So I'm gonna, all this work that I showed you today is the work of one of my grad students who's co-mentored by Noah Zeitlin, Angela Way. She is currently in New York trying to validate some of this work in Emer Kenny in BioMe. BioMe? Yeah, in BioMe at Mount Sinai. And so she was hoping to be here this year, but hopefully next year she'll be here to share some of her work. And I'm happy to take any questions.